Thank you, Prime Movers family, for joining us today. I am quite confident that this is going to be one of the best webinars yet. It is a privilege and an honor to bring you all together today to meet with these amazing gentlemen that I, through my career, have learned from and known for a while and considered a mentor along the way. I think with synthetic biology, with the combined brain power and Rolodexes of these three gentlemen, you probably have the entirety of the industry one step away. Michael Clay, who I actually did a little bit of my graduate work with, has worked in almost every synthetic biology company that matters. Todd Peterson is a juggernaut in the industry, being CSO of a number of amazing places. And when politicians want to know about synthetic biology, they call Rob Carlson. Any of you that have read a little bit about synthetic biology may have come across the Carlson curve. This is that Carlson that we're talking to. And speaking of talking to them, that's probably enough talking to me. And I will let them introduce themselves far better than I could. But before that, just one little housekeeping item. This is an interactive fireside chat. Please put in the chat the questions, comments, thoughts that you want, and we'll fold them into the conversation. Thank you very much. And with that, Mike, please give us a little bit of a background.
Thank you so much, Caleb. Really grateful to be here and connect with everyone. So like Caleb said, I feel like I collect synthetic biology companies. I've worked from Codexis all the way to Inscripta, and I'm now the co-founder and COO of a company called Infinome Biosciences. And so Infinome is it's a contraction of the two words infinite genome, and it represents the infinite genomic opportunities that we want to bring to the market. And so we're a next generation lean bioengineering company that engineers biology faster, efficient, and cost effective to reduce risk to bring products to market. So thanks a lot. Great to be here.
Todd, would you mind giving our audience a little bit of background on yourself?
Sure. My name's Todd Peterson. I have a background in molecular genetics, and I've been in biotech industry at medium, small, startup, and large global organizations for most of my career. Relevant to synthetic biology, started in the field in the mid 2000s, really, through connections with SINBRG, which was a National Science Foundation virtual center that probably arguably much of the synthetic biologists and synthetic biology companies are in some shape or form derived from that organization. I was the head of synthetic biology R&D at a company called Life Technologies, which is now Thermo Fisher, which was really trying to make synthetic biology easier and much more predictable from a tools standpoint. And thereafter, then I went to synthetic genomics. I was CTO there for 5 years. Most recently, I was up in Seattle, up at the Allen Institute as a chief scientific officer, and I'm back down now in Southern California, and I'm doing full-time advisory work and board work and so forth as the founder and principal of GenApex Bio.
Thank you very much, Todd. And sticking in Seattle, Rob.
Hi, I'm Rob Carlson. My day job is the managing director of Bioeconomy Capital, which focuses on early stage seed ±1, we like to say, investments in tools companies, tools and manufacturing companies, although we're pretty flexible. We led a B round in one of our companies when it was an obvious thing for us to do. And we really want to build out the infrastructure to be able to do real engineering, predictive engineering and manufacturing and biology. And so we were early investors in Zymergen, for example, to that end. They're now public, obviously. years ago, I wrote a book about how I thought all this was going to play out called Biology as Technology. And the core of the thinking in the book and also of our practice at Bioeconomy Capital is being as quantitative as possible about trends. So I've focused on quantifying how fast technologies are going, how well they work, and then also how much money there is in different parts of the bioeconomy. We can talk about some of that as we go along.
Thank you. Thank you very much. This is going to be a very exciting conversation. And again, I welcome as we go, please populate your questions in the chat and we will fold them in. Maybe we can just start off. We all on this call know what synthetic biology is. And I, for one, believe that synthetic biology is the next industrial revolution. But maybe we could start at the back and just define what synthetic biology is in your opinion. And maybe Rob, you could kick us off with that.
Well, to be honest, we should definitely talk about synthetic biology, but I was reflecting on this before the call and I'm kind of tired of the conversation about the name. I guess I should say what I think it is first. And what I think it is applying engineering principles to biology, being able to build stuff that behaves the way you predict and can be used for manufacturing and healthcare and every other application that biology is clearly a part of, as well as many that we will discover as we go along. Biology can do amazing things that we have yet to really get our hands around. And I say I'm tired of the conversation because I go back to before when it was called synthetic biology. And in the late 90s, when Drew, Andy, and I were at the Molecular Sciences Institute trying to put our heads around, describe what this thing was that was coming, we knew we didn't want it to be called genetic engineering because Monsanto had at the time commandeered that term and was misusing it. They weren't really doing engineering, as exemplified by the fact that their products didn't behave as they said they should. Crops failed and different problems here and there. So we wanted to distinguish this new thing from what was going on, and we wanted it to be about real engineering. And so it was called a number of things. We tried them out, and then we were at a cocktail party that Nature threw in San Francisco when they opened their San Francisco headquarters, and Carlos Bustamante said, you young kids, this is obviously called synthetic biology after synthetic chemistry, et cetera, et cetera. And so that's what we settled on. And, for good or bad, it's called synthetic biology. It doesn't really matter at this point. Call it constructive biology, call it bioengineering, biological engineering. It's just about using the tools we have to engineer to make stuff that behaves the way we want and improves the human condition and hopefully makes the planet greener and easier to live on as we go.
Really great point. And in one sense, there's nothing synthetic about it, right? It's really just using nature to build things that are of value to us. Todd, diving in on that, where do you see the things of value that biology can really build for us? What of your career that you've been working in large companies and mid, what are the kind of big areas, big places that we should be thinking about leveraging this nature as a workhorse?
There's many applications for synthetic biology as a broad platform for engineering of living systems and their components for useful purposes. That's the operating definition that I use. I think in the beginning, just because it sort of needed to start in a reasonable place, that there was quite a bit of attention that was focused on essentially prokaryotic organisms, one that were pretty well established and well understood, and for which there was sort of a nice set of tools where there were a number of strains where the genomes were sequenced. It was very clear how to basically handle those. And some of the early work really was in essentially leveraging those in fermentation mode to make various products, but primarily biofuels was very popular in in the beginning, a number of companies kind of came and went there. But, you know, I think what's happened is that the tools have really gotten much better, much more universal, that the level of knowledge, not necessarily understanding so that it's absolutely useful for all forms of forward engineering, but just in terms of the basic understanding and the interchangeability and how one takes parts, for example, that is, for example, genes or circuits from one system and essentially has some function in another. So the level of engineering is getting more sophisticated. I think the number of systems that are available has started to branch out from things that are very well characterized to more novel types of cells. I think there's been a big shift. It still has a ways to go really to get to more sophisticated engineering. But I mean, we're seeing this really in gene therapy and cell therapy as you're stepping out from, you know, commodity products or, you know, green chemicals into more therapeutic applications.
Thank you. And building off that, we actually have a question from the audience already that I think is tailor-made for you, Mike. It says, assuming that you have automated robotic lab and all the algorithms that you need to iterate through tens of thousands of enzymes, which we kind of already have these. It's not much of an assumption. The speed is debatable, but we do have those technologies. What are the most high inspiring and sci-fi level applications that you would target?
Oh, the targets, I would say I guess enzymes or genomes, targets that are maybe in the protein space. I think all of the areas, Rob has done a nice job kind of categorizing out the areas of opportunity being in pharma, ag, and industry. And so if you had a platform that where you could crank out tons of small molecules or natural products or peptides or proteins, I mean, I think that would be pretty broad across all those areas. So I don't know if that answers the question, but I guess at the highest level. But we can drill down into his question a bit more, her question.
What are some specifics? Where would you focus on? Would you be making textiles? Would you be making pharmaceutical drugs? Where would you put your time and money?
So I guess at Infinome, we're pretty broad in terms of like what we want to go after. I don't think there's any thing that is off the table. You can see, like, at least in the space out there right now, in pharma, you know, if you wanted to, maybe there are better ways to make antibodies or peptides, CRISPR gene therapy. In ag, there's cryotoxins. All those are proteins that need to be produced. These products in industry, flavors and fragrances, you see on the kind of the food side, you see players like Impossible Foods making leg hemoglobin and milk, Perfect Day also making milk, Clara Foods making egg albumin. On the material side, there's Bolt Threads and Modern Meadow that are producing various products for like clothing or textiles, flavors and fragrances, cosmetics. Geltor is making collagen. I mean, I could go on and on and on. I think that for Infinome, we're kind of open to all of those spaces. And then I haven't even talked about the small molecules. There's lots of examples of companies creating small molecules, such as Codexis has worked with Merck to engineer enzymes to make the compound Januvia, as well as Amyris with artemisinin. As you guys know, the psychedelic or the cannabinoid space is big right now. You have a lot of players in that, like Willow and Demetrix and Amyris and a variety of companies. In ag, there's biopesticides. I could keep going and on and on and on. A lot of opportunities. There's more opportunities in SynBio than there are companies to address them. So I'll say that.
So it sounds like the shorter list would be what places this couldn't work. Maybe almost a zero list.
Actually, Caleb, I think that's a really good point in that we don't want to dismiss chemistry, even though the synthetic biology people would love to replace it. I think that it complements chemistry. So maybe there's a lot of products that chemistry can't make. So you're not going to see a synthetic chemist make a protein, or maybe a natural product. They could make that in a lab, but to commercialize it is maybe too many unit operations. So that's where you want to leverage biology. But then once you make those products, it's possible that they could be decorated in various ways by chemists. So you really want to partner with whoever you need to bring these products to market.
And Rob, it's a similar question to you. I mean, you've put a lot of your money and other people's money to work across a number of companies quite successfully. Where in the next couple of years do you see the highest impact opportunities?
We tend to look at that in a couple of different ways. So one is how quickly can we get to market? What are the overall costs? Is it regulated? Is it not regulated? So obviously, if you're going after a pharmaceutical product or frequently a crop, then you're facing regulation. So then it's a matter of, do you want to take that on? How fast can you do it? Our thoughts there are informed in part by the fact that I used to sit on a National Academy of Sciences committee whose job it was to teach regulators about synthetic biology and about new technologies. And that was always a challenge. And so when it came time to starting a fund, I was terrified by that prospect, frankly. I didn't want to take new technologies to the FDA. Now the FDA has changed a bit, and so we're more open to going after drug-like things. Anything that faces a patient, we're still very careful about that, but that's a possibility. Another way to think about it is margin. So, a barrel of oil is not a homogeneous thing. Chemicals are not the same cost, the same functionality, the same market size. Only about 2% of a barrel of oil goes into petrochemicals on average. There's 2% of the volume of the atoms. And even there, the cost per kilogram ranges from $1 or so to $1,000, could be $10,000 a kilogram. And then you have to be able to, if you're going to participate at the $1 a kilogram, couple dollars a kilogram scale, you need to be able to get to 100 million kilograms quickly in order to make $100 million in revenue. So that's a big challenge. And now you're talking about not just the biological engineering, but also steel in the ground and the process engineering and where you get your feedstock from. And that's not a small amount of stuff to move or to sell. We have typically focused on the other end of the value chain. So other end of the value spectrum, sorry. So small volume, very high margin, very high price. And what has made that easier is that now you can build enzymes that accomplish chemistry that's never existed before. So whereas synthetic chemistry has a spectrum of molecules that you can reasonably expect to make in a petrochemical facility, there's a whole mess of things you cannot make or cannot plausibly make in that facility that we'd like to have access to. And enzymes are magic. You can make that stuff using biology, and it has improved material properties. It can have improved economics, can be more recyclable. And so that's where I'm most excited. Both Zymergen and our Zeta from our Fund One are working exactly down that road, and they have come out with products that you cannot plausibly make using petrochemistry, and that perform way better than anything you can get out of a barrel of oil today. And then that would go to my final point here, which is that one of our unspoken theses, or one of our unspoken desires with all of our activities, is to kill oil as quickly as possible. It's hard to have that be an investment thesis because oil is gigantic. It's not going away anytime soon. So you judiciously have to decide, can you, with this money, move the needle in any way on taking market away from oil or outcompeting oil or whatever it is. And now that we've laid the groundwork with the engineering tools and some of the manufacturing, although there's a lot of work left to do there, we can start asking now, how do we use those tools to go after displacing oil from the world. And we just made an investment in our first company that I think is the first time we've seen a commodity chemical play that we thought made any sense. And so, you know, as the technology has evolved, as the markets have evolved, it's become easier to figure out how to make those bets. But then also we have a different financial infrastructure, a different financial conversation than we've ever had before. You see companies pulling back from petroleum. You see banks deciding that the return on capital and petroleum companies is no longer that interesting. Where else can they put their money? Well, here's a place you can put your money where you can outcompete petroleum, and at the same time displace, you know, that enormous carbon load from the planet. So those are the kinds of things we think about.
Interesting, interesting. Maybe I could pick up one of the things you mentioned there, and also grab a question from the audience here for you, Todd. What are the big challenges? What are the limitations? What are the things that are kind of preventing this industry from achieving the potential we all believe it can?
Well, there's probably a couple things to mention. You know, the first is that although the tools have gotten better, and in general, I mean, the knowledge has certainly increased, and the cycle times through essentially high throughput strain construction and kind of acceleration of the so-called design, build, test, learn cycle and application of machine learning and you know, artificial intelligence to, you know, massive amounts of data. That is that is accelerating the cycle. But you know, there's still actually quite a bit that we don't know. I think particularly, you know, as it relates to this, depending upon the organism, very highly evolved space, essentially the interface between the primary metabolism, which is what the organism needs to basically grow, and secondary metabolism, which is what the organism takes from primary metabolism in order to make the products. And depending upon the product, as well as the organism, as well as the process that's involved, you know, all of those things are a little bit of trial and error still. So I think it's not right to think that the state of the technology is at a point where it's like sitting down and you're looking at a 777 jet or you know, making a bridge where you know, there's sort of uniform physics and there's uniform principles out there. There is still a fair amount of trial and error, but I think those things will eventually and much more rapidly, through larger data sets and application of intelligent you know, use of data, that the principles and the algorithms are very likely to fall out of that. I think there's another thing, which is that it's not fully appreciated in many cases what the risk is and what the difficulty is when you basically design an organism. It works fine at small scale, but now you have to scale it up. I think that is more true in these, you know, well-established systems like yeast and others, where it's much more predictable what happens when you get to you know, 100,000 liters or more. For novel microbes, very few of those have actually gone through that cycle. So there's always going to be new things to learn when you essentially try to scale the technology, which, as you look at most of the products out there, many of them are absolutely requiring scale and are you know, low margin commodity products. So, you know, there's a lot to learn there that's in this kind of gray zone that has to do with what you engineered, the way the organism grows, the primary metabolism of that organism under scaled fermentation conditions, and how that feeds into the actual biosynthesis and the chemical production of your product.
Yeah.
Yeah, and we've, I mean, we've been brewing wine and beer with yeast for quite some time. That could arguably have been the first synthetic biology product. We talked about this a little on our pre-call over the weekend. And maybe I go back to you on this one, Rob. Yeast and E. coli are kind of the most abused and well-known organisms to produce things, but there are a bunch of others coming online. I know one of the companies in your portfolio uses microalgae. How do you think about these, and where do you see these new organisms, or chassis as they're called in the industry, playing into the whole of synthetic biology as it's maturing?
That's a great question. There are a couple of things I'm quite excited about. The first is that, as good as E. coli is to make some things, it can't make everything. As good as yeast is to make some things, it can't make everything. So Sarah Richardson, who started a company in our portfolio called Microbiome, likes to say, why would you, if you want to make milk, why would you take a dog and teach it to eat grass to make milk? No, you go to a cow, which naturally eats grass, and you figure out how to help it make milk better. So, Microbiome is focused on exactly that, matching the bug to the problem at hand, rather than forcing the bug to make something that it doesn't usually make, or eat something that it doesn't usually make. There's a diversity of organisms in the world, diversity of microbes in the world. So one thing that's coming is we will be choosing better platforms to do the manufacturing that are more amenable to that kind of manufacturing. So there will be a diversity of chassis of organisms. There will also be a diversity of manufacturing systems that result from that. So the microalgae company is Lumen here in Seattle, and we invested in them because they looked like an industrial manufacturing play to us that could be photosynthetic. So that was orthogonal to everything else that existed. And then it turned out that via a chance conversation with the Gates Foundation, that they can make antibodies for about 1,000-fold less than any other platform around. And the magical part of that is that they can grow this, and first they can engineer this bug, which is already food grade. So you can make the antibody and then eat the organism and then receive the therapeutic benefit of the antibody, which is a huge step forward. But then also they can grow it indoors in photobioreactors. So this basically is a manufacturing system that you can, biomanufacturing system that you can plug into the wall, plug into the grid, and it is fed by electrons rather than sugar. So that's another thing that's coming. And then the next step, which is harder and will take longer to play out, but ultimately embraces the way that manufacturing works via biology on the rest of the planet, and that is ecosystems. And if you have a diverse ecosystem, it is far more productive than if you have a monoculture. And this is true whether you're growing corn versus diverse crops, or whether you're growing a single microbe versus diverse microbes. The trick is the diverse system may not behave in exactly the way you want at the beginning. You have to learn how to coax it into doing the right thing. And the best example of that we have in our portfolio, and we share with Prime Movers Lab, is Upward Farms, where they are basically doing ecosystem engineering. And again, this is in a box, it's inside, it's plugged into the wall, and it's this crazy productive manufacturing system. And while the organisms themselves have not yet been engineered in that system, in our view, what Upward Farms is doing is not merely selling lettuce and selling fish. They are demonstrating ecosystem engineering powered by electricity, and ecosystem level manufacturing powered by electricity. And inevitably, that will also include engineering the organisms, again, to accomplish manufacturing of substances at scale that we can't even imagine doing right now. But first we have to get the engineering technology and the manufacturing technology right. And that's being demonstrated.
Interesting. And inside, encapsulated in that, I heard, biology is complicated. Sometimes it doesn't do exactly what you want, and sometimes that's fortuitous. Luck favors the prepared mind. Digging a little deeper on the tools, and Mike, you've had a lot of experience at a bunch of the various startups working with the different tools like CRISPR. And what do you see with these new robotic and new genetic and the new tools that have come online in the last couple of years? How's that impacting the industry, and what are the opportunities or new opportunities it's creating?
Yeah, thanks, Caleb. Yeah, I to kind of combine Rob and Todd's answers together, I agree with them that engineering biology is just currently, it's slow, it's laborious and expensive. It's just how it is, and it's that way because of these tools are just starting to kind of come online. So it's not enough to just do a little bit of CRISPR, but rather high throughput CRISPR. So having those capabilities, having access to that, and the companies, when you look across the space, companies that don't have those tools or if they're in organisms that don't have tools, it doesn't mean it's not going to work. It just means it will take a bit longer, or quite a bit, could take a long time to bring things to market. That's kind of how it's been in the past, but I think those are changing. So the tools on the design side, you're starting to see a lot more massively parallel genome scale precision engineering. And then on the build side, the high throughput CRISPR to create a lot of diversity and to sift through that and to recombine that diversity. These combi libraries are very powerful to make large leaps in fitness space. Companies that have these tools have an unfair advantage against those who don't. Same thing with...
What quickly is fitness space?
That's a good question. It's really just any phenotype, anything that you're making, any product people want at scale, high titers, high yield, high productivity. So usually what happens is you'll see people that go into the lab and they can take genes or pathways from other organisms and drop them into, they can either use those organisms. Some of them are recalcitrant. They're very difficult to engineer and people are kind of stuck in them. But now we have the tools to take those pathways out and to drop them into, let's say, E. coli or yeast is a possibility. And then once they're in there, people are making these products, but maybe at a really tiny scale, maybe like micrograms or mgs. But in order for them to be commercial, they need to be at the gram or kilogram scale. So in order to get from that microgram to a lot like kigs, you need to go through these. The aspiration is that we want biology. We want to approach biology in an engineering mindset. Is the aspiration that we can just drop in these genes and everything goes to plan? But it's not like that right now. It's not like computer science or any of the engineering fields that we know. It's biology is a lot more difficult, as we were all kind of talking about. And so the way that most of these companies get through that is to go through these, also another engineering approach, which is design, build, test, learn through these cycles. And then as you go through them, you increase the tighter productivity and yield.
And so just to clarify, the potential products that synthetic biology or nature can make, is that restricted only to the products nature makes today? Or do we have the ability to expand and develop analogs and go out into a larger space and do things that synthetic chemists do, or do things that synthetic chemists cannot even do today?
Absolutely. I don't think anything's off the table in terms of there's creating what's already been made, but then there's also altering or engineering the organism to make all kinds of a diversity of products that nature hasn't created before. And I think that's also pretty interesting and also maybe good for humanity in a variety of ways, and also maybe good returns for investors. It's a whole area that's just kind of opening up, and it's gonna open up a lot wider as the tools kind of become more available.
Thank you. Todd, maybe similar question here, but could you give us a little bit of the large company perspective on that, on the tools and how we're looking at the space in synthetic biology?
So most of the larger companies, I think, have started on the sidelines, kind of what is this curious thing, synthetic biology. I think it's at a different point now where larger companies are starting to get into into the game, and they're interested in kind of targeted ways for essentially coming to synthetic biology with specific classes of products in mind, and they're primarily doing that through a partnering type of approach. Some of that's M&A, some of that is more kind of traditional partnering. There are a few large companies that have put kind of stakes in the ground and have started to build their own franchises with respect to tools. There's probably a lot of those out there. And there's a few of the larger companies. I'm the one that kind of pops to mind right now is DSM, who have made a significant investment in the core technology and are taking a hybrid approach where, I think that they have quite a bit of capability at this point, as well as a very aggressive partnering strategy.
We on this, we got a question from the audience here from Scott. Do you see strategic partnerships as very complementary to the capacities of the companies and paramount to success as it kind of plays out in biotech? There's a very, you want to see these companies partnering early, or do people go it alone and bring their products to market themselves? How do you see the field?
I think that their complementary approach is probably the fastest and is probably the least risk and it's probably, the better path. For example, in the therapeutic space, it's pretty tough to be fully, vertically integrated and have, as a small company, all the regulatory expertise, all the manufacturing, all the sales, marketing, sort of distribution, all that stuff that big pharma is much better at than kind of the very, very front end, the raw, kind of raw discovery and let's say technology innovation pieces of it. So that's the best type of partnership. You see it also, and there's a number of examples of this where there's smaller companies that are more on the synthetic biology side, on the biological engineering. They have their own platforms, they have their own set of tools, but they don't have the scale up and they don't have the distribution. And as Rob pointed out, steel in the ground. And this is expensive. I mean, to build a scaled fermentation plant for a new commodity chemical like that is hundreds and hundreds of millions of dollars. So you see a lot of partnering there as well.
Interesting. And that actually leads into the next question we got from the audience, which is targeted at you, Rob. How do we solve the fermentation capacity crunch at the pilot level? Why aren't people building new capacity for non-pharma biomanufacturing? And I'm going to paraphrase this last part. How much are you talking to Joe Biden about his infrastructure bill and helping to support this?
In reverse order, not very much. I'm taking pot shots out in public, but no, they haven't asked me for specific help on this.
How do you think? Do you think the infrastructure bill will help this?
It could, but this gets actually to the first part of the problem. It's not merely about building fermentation capacity. It's about putting all the process management stuff in place so that you can use that capacity. Mm-hmm. And I don't know that we're actually short of fermentation capacity at the moment. There's a huge amount of ethanol manufacturing capacity out there that's going unused, is mothballed for economic reasons. Who was it? Gevo tried to walk in and.
Point was, so the part of the question that kind of stood out to me there, sorry to cut you off there, Rob, is fermentation capacity at the pilot scale, right?
Right. Oh, that's true. At the pilot scale. So, again, I'm not certain that we're actually that short of it. We could be, and it doesn't cost that much, I think, to get into the pilot scale. The harder problem that I see companies facing is from pilot scale to getting into the marketplace. And there, the technical problem you have is, how do you do technology transfer? How do you do process transfer? How do you define the process wherever you are so that you can take it over to this other facility and boot it up? This is also true if you're, at a few liters or maybe even 100 liters and you want to go to 10,000 liters. Call that pilot scale maybe. The core problem is not, do we have enough 10,000 liter fermenters around? The core problem is, how do you walk into where the 10,000 liter fermenter is and get your bug booted up and running and making the thing you want? And the assumption frequently that startups have is that they can just say, here's our bug, here's how we grew it, do that for us. And the problem is that the people receiving that, the contract fermenting operations, the contract manufacturing operations, they don't have knobs to set that are amenable to the process description that the startups show up with. So this was a huge problem in semiconductors until 1980. And it was solved by ironing out this scale-up issue by making it possible to have a design that could be implemented anywhere, could be compiled to anybody's manufacturing hardware.
Mm-hmm.
And we have to have that same infrastructure in biology. I think that's what we're investing in. So we invested in Riffin and Synthase to accomplish that kind of a scale-up transition. And it does work. It's also very early, and not everybody is thinking in the same way, let's say, about that problem. So there's still very much, I think, a prevalence in the industry that you do build a process and you build hardware around that process in a bespoke fashion, or you start with hardware and you build a biological process that goes into that hardware in a bespoke fashion, rather than they're decoupled in some way and you have the biological process and a description of that process that you can walk over to anybody's hardware and just get it started. We don't have that, and we got to get that somehow.
Yeah, and a good point that you mentioned on too, that it's different for what scale you need to get to depends upon what your end product is, right? And the biofuels, that's one of the largest scale things you could imagine, but some therapeutic entities, or as you mentioned earlier, the high margin, low volume products, that size scale up may never be needed to have a profitable organization and entity.
Yeah, that's a really, in fact, to kind of jump off that little, I like what you were saying, Rob, and I kind of wonder if right now there's just tons of chassis out there. There's all kinds of hosts. There's filamentous fungi for making lots of protein, or CHO cells for making protein. Then you have like streptomyces for making natural products, and you have all these like different bugs, Pichia, and for also making protein. And what we're finding is like all of those bugs need to have kind of special fermentation facilities for them. And I kind of wonder if we were to kind of have that VHS Betamax head-to-head, and kind of maybe eventually we would collapse down to like a handful of strains that use like all the same type of fermentation facilities, then we can kind of leverage some synergy between all of the different synthetic biology companies, and that if they were in some more kind of common hosts, that they could use some of the same scale up facilities, maybe make things a little easier, rather than to have all these like specialized hosts that also need these specialized tools that need specialized fermentation and scale up. And maybe that kind of, maybe over time, if we have that consolidation, it might help bring even more products to market.
I want to riff on that for a second with the observation that it's early still.
Yeah.
And one of the things people get confused about in biology is that they compare it immediately to airplanes or immediately to chips, and without kind of working through all what came before, right? And so what came before in chips was math and physics and material science, and building that capacity up from zero so that it works according to a particular description. Yeah. So that means, if you're a software designer, you sometimes forget that the chips are built to work in a particular way with particular rules that have to also to do with what's manufacturable.
Yeah.
and they're delivered to you and they function and you operate within that environment. Similarly, if you're an electrical engineer and you're making a new chip, well, you have constraints upon your ability to make stuff that has to do with manufacturability and what's already been scaled up. So you walk into this world where everything works because it's been built to work, and you can do a thing and it also works. And in biology, you walk in and you pick up this stuff that's, you know, slime laying around on the ground, and you have to figure out first how to force it into a particular shape and then make it do your bidding. And so we don't have that same kind of infrastructure in biology. We're having to build it all. And that leads me to the fundamental point there, which is that we're trying to engineer biology as we find it. That's going to be quite different from when we can engineer biology as we build it. And we're still working on that part of it. And you can see particular examples where it's working really well. So Arzeta, for example, can design enzymes with brand new chemical functions that have never existed before. And they can do that in a predictive way because their design tools are based on physics. So they take an enzyme from nature that does a particular thing, and they say, that looks like a good candidate to do this other thing that doesn't exist. And we're going to start moving atoms around by half a diameter to make new chemistry happen. And it works. They can make enzymes jump through hoops like nobody's business. And they can use those enzymes to accomplish unit chemical operations that, so far as we know, have never existed in nature before, to make compounds that have never existed in nature before. And again, that you can't plausibly get to by synthetic chemistry. So now we're really building biology from the bottom up based on physics, but this is a brand new capability. This is not even, 2010. This is 2020, 2021. And the world is opening up to build things from scratch in a way that it never has before.
I completely agree. And one of the things that I find a little daunting, but also the opportunity, in order to build these things, in order to design it, we have to be able to isolate and culture that organism from it. And still, I think we as a society can only isolate and culture 10% or less than 10% of the microbes that we know of. So the industry that we're talking about today is built on 10% or less of the potential capacity, and that's in the natural world. And as you're mentioning, Rob, we can even build beyond that. So you can take that other 90% and expand that pie even more. And I think, to me, just from there, the possibilities are limitless of what we can do, what we can build, what we can find. And the beauty of nature, and to use one of your terms, Rob, is circular economy, is that unlike bioindustrial processes or chemical processes, nature usually does this in a very efficient, minimizing pollution, minimizing waste fashion, and creates really just one or the product that is wanted at the end of it. On that topic,
Let me make a point there if I can, because I think that there's a place, there's a very good place that is not quite as predictable as it needs to be, but it will soon get there, which is that you can basically go just to the environment and sequence pathways and genes and genomes at that point, and not even worry about actually cultivating those particular microbes. I mean, if they have a very interesting pathway or a set of genes, very often, and this is also improving, you can do essentially informatic analysis and put those things together, at least provisionally, to get some idea about what would be the underlying chemistry that is produced. And then you take those out of those organisms. You don't have to grow those organisms. You don't have to figure out what the secret sauce is to get those particular genes to turn on and so forth. You take the machinery out and you engineer it so that it's at your design and at your command.
We've got about 10 minutes left in our panel. I welcome the audience to please throw in the last questions that you have. Otherwise, we're not going to get to it. I'm going to ask my favorite question of each of you panelists now. It's a twofold question, and we'll start with you, please, Mike. It's the prediction. It's the contrarian question. What do you think will happen in this industry in 5 to 10 years that people today would say you're crazy? That's part one. And part two is, other than, and for you specifically, other than Infinome Biosciences, what do you think the best investment opportunity is in this industry right now?
Oh, good question, Caleb. So I think that so the questions around the next 5 to 10 years will come to fruition that most people would disagree with. I would say, I don't know if they would disagree. I would say the future is people being able to do far more with fewer resources and people. Like, so there's that. I think another aspect of that is right now, sequencing, like reading and writing, or sequencing and editing is cheap enough that by themselves, I haven't seen like the widespread success in the bioeconomy just from like reading and writing coming down in price. And so it's more than just that. You know, it's more than just reading, just saying that these prices are going to fall and the heavens are going to open up and all these bioproducts are going to fall out of the sky. It's also, you need to have something a bit more. You need, the tools by themselves are not sufficient. You need to have the strategies and the people with experience to kind of push them to commercialization, to enable them. So I think I'm going to, and so who would I invest? I'd probably invest in companies like maybe our parent Inscripta that are like offering those tools that are democratizing those powerful tools of, they have a CRISPR nuclease that people can access. It's not like Cas9, it's MAD7. You know, offering that ability to do high throughput genome editing to go faster, easier, cost effective. I think that's, you know, really, it de-risks everything and allows more products to come to market.
Rob, same question to you, please.
Right. So two crazy things. One, I said my day job is running Bioeconomy Capital or managing director there. The other thing I do is that I'm a affiliate professor of computer science at the UW, working on this DNA data storage project with Microsoft and more broadly molecular computation. And when they asked me to start that, I thought it was a bit nuts, bit of a lark, you know, a good consulting gig. And then very rapidly, I realized that not only could it work, but it was going to work and that it must work. And so if you look at how much information we produce and how much information we need to store, it is outrunning our capacity to make and store tape, physical tape or hard drives. And there isn't a plausible, on the 5 to 10 year, well, maybe beyond 10, 15 year horizon, there isn't a plausible way to imagine storing all that information on the media that we've sort of grown up with. We have to jump off that curve onto some completely different curve. And the only thing that I can see right now is DNA. We can read it, we can write it, we know it lasts for 1 million years in the environment. So let's do that. And you know, if you look at the number of atoms per bit you've required to store that bit, there's nothing that beats DNA. So I think we're going there. The next thing that's sort of crazy that people probably disagree with is I think we're going to redo most of molecular biology over the next 10 to 20 years and do it with a focus on quantitatively small error bars. So rather than I made this measurement and the thing turned green or not, it's going to be I made this measurement of activity with 1% or this measure of affinity with 1% error bars. The reason I say that is that within our companies, we see that getting down to 1% error bars is the critical step to enabling predictability. And if you're above that, then you don't really know what the hell is going on. Your models don't really describe the system, and you have to accommodate through resilience rather than actually doing real engineering. And sure, things should be resilient, but on the other hand, you want them to be predictable. And the way you get predictability in every other field of engineering is with quantitative measurements and small error bars. And I think that we're going to discover that much of the biology we think we know is wrong. and that we're going to get it right by measuring it better. And who would I invest in? You know, I don't know. I am struck every day by the extent to which it's now quantitatively true that when you install better engineering tools in companies, they get quantitatively better. So Riffin, for example, I mentioned this before, they now have quantitative results from Novozymes and BASF, where those companies, which are, I mean, innovation is their core to their identity, and the fact that they're big companies, they doubled their product development and commercialization rates using Riffin. Or Synthase, you know, you drop in this automated system to be able to do experiments and analysis and control entire laboratories using this operating system. Then you get, you know, 10x here and 5x there, improvements in cost and productivity. And these are fundamentally transforming the companies that use them. And whether you invest in those companies or some other company that is able to change the way, fundamentally the way that research and development and commercialization works in biotech, that's why we exist. And I'm all in on that still.
Thank you. And Todd, take us home with your predictions, please.
I fully agree with the glimpse into the future there. Some of the things I think that there's going to be cell therapies and cell engineering for human disease, complex human disease, that will be much more sophisticated than you know, sort of the current versions right now with CAR T and other types of relatively simple, let's call it genetic manipulations. I like this idea, and there's some regulatory challenges here, but I would say that in general, as you move up the curve with respect to engineering sophistication, sensory and response, making cell therapeutics that are really smart cells and can do more than just one thing. The next version of that, which is around the corner, is to have these, you know, 2 cells in a population, or even take that further and start to do the engineering of tissues. That could be entirely bio-based, or that could be a combination of cells and you know, synthetic biology and engineered cells in you know, various types of structures or scaffolds or various devices. I think that's going to be very exciting to see how that piece of it actually comes out. I think for sure there's going to be a maybe second or maybe even third cycle of small molecule discovery, which is based upon synthetic biology and natural products. The first version of that was just natural strains. Second version was, you know, combinatorial biosynthesis. And I think synthetic biology is going to be very much going to put small molecule drug discovery back, you know, into the forefront after pharma basically abandoned that many years ago.
Thank you. I completely agree with that. I think that synthetic biology is the next manufacturing wave. I've definitely drank the Kool-Aid that you 3 gentlemen have fed me throughout my career. So I also thank you all for that. It is just super exciting what things are on the horizon. And I think I'm looking forward to seeing the predictions you 3 made come true. We at Prime Movers are looking forward to supporting those and supporting the entrepreneurs and innovators that bring them forward. And I just think that synthetic biology, with the definition and the points you made earlier, Rob, is really where the next generation of massive value globally will come from in the next 5 or 10 years. Thank you all for joining us in our webinar today. Thank you all to the attendees for spending your time with us today and learning about this fascinating field.
Thank you.
Hey, thank you.